beginning with Module 2.3, the visual pathway, before continuing with the text. We know a lot about the underlying systems that support vision in comparison to other systems. Rather than consisting of one pathway, the visual system consists of multiple parts which extract elements of information from the environment and send these to the brain for processing. As shown in Figure 5.1, there are a number of processes that occur from the time a stimulus is received to when it is perceived. And the figure, I think it's in the book, is basically just a labelled arrow starting with sensation and showing the process. So the first process is stimulus is received by sensory receptors. Then the receptors translate stimulus properties into nerve impulses, which is called transduction. Feature detectors analyse stimulus features. Then stimulus features are reconstructed into neural representation. Neural representation is compared with previously stored information in the brain, and matching process results in recognition and interpretation of stimulus, ending in perception. The nature of light. If we start to consider the visual stimulus itself, it is electromagnetic energy, which is light, measured in nanometers within a particular spectrum. The human eye can only see between the band of 400 and 700 nanometers. Bees can see ultraviolet light, and snakes can detect heat and infrared energy. Figure 5.6, light energy, on the next page of the e-text, shows the full spectrum of light energy and how narrow the human vision truly is. The visible spectrum that we can see depends on our eyes. However, we now have technology that transposes information from one spectrum to another. As a result, there are going to be a number of assumptions that may not be accurate, as night vision goggles or heat may change what we perceive and impact our overall sense-making. Major structures of the eye. The eye processes light and passes this information to the brain for processing. The eye consists of the following key parts: cornea, iris, pupil, lens, muscles, retina, blind spot. Now back to the e-text. Vision. The normal stimulus for vision is electromagnetic energy, or light waves, which are measured in nanometers, nm in brackets, or 1/1000th of a metre. In addition to that tiny portion of light waves that humans can perceive, the electromagnetic spectrum encompasses X-rays, television and radio signals, and infrared and ultraviolet rays. Bees are able to see ultraviolet light, and rattlesnakes can detect infrared energy. Our visual system is sensitive only to wavelengths extending from about 700 nanometers, which is the red end of the spectrum, down to about 400 nanometers, which is the blue-violet. To remember the order of the spectrum, from higher wavelengths to lower ones with the name ROYG-BIV, ROYG-BIV, red, orange, yellow, green, blue, indigo, and violet. The human eye. Light waves enter the eye through the cornea, a transparent protective structure at the front of the eye. Behind the cornea is the pupil, an adjustable opening that can dilate or constrict to control the amount of light that enters the eye. The pupil size is controlled by muscles in the coloured iris that surrounds the pupil. Low levels of illumination cause the pupil to dilate, letting more light into the eye to improve optical clarity. Bright light makes the pupil constrict. Check out the diagram on page 152, which is figure 5.7. Behind the pupil is the lens, an elastic structure that becomes thinner to focus on distant objects and thicker to focus on nearby objects. Lens is also a key term. Just as the lens of a camera is used to focus an image on a photosensitive material, film, so the lens of the eye focuses the visual image on the retina. Retina is also a key term, and it is described as a multi-layered light-sensitive tissue at the rear of the fluid-filled eyeball. As you will see in figure 5.7, the lens reverses the image from right to left and top to bottom when it is projected upon the retina, but we perceive the image in the correct orientation. So that's really cool, because I've always heard that we see upside down and the brain flips it. The ability to see clearly depends on the lens's ability to focus the image directly onto the retina. If you have good vision for nearby objects but have difficulty seeing far away objects, you probably suffer from myopia, which is short-sightedness. In short-sighted people, the lens focuses the visual image in front of the retina or too near the lens, resulting in a blurred image for faraway objects. This condition generally occurs because the eyeball is longer, front to back, than normal. In contrast, some people have excellent distance vision but have difficulty seeing close-up objects clearly. Hyperopia, long-sightedness, occurs when the lens does not thicken enough and the image is therefore focused on a point behind the retina or too far from the lens. Eyeglasses and contact lenses are designed to correct for the natural lens's inability to focus the visual image directly onto the retina. Photoreceptors, the rods and cones. The retina, with its specialised sensory neurons, is actually an extension of the brain. It contains two types of light-sensitive receptor cells called rods and cones. Because of their shapes, see figure 5.7b, there are about 120 million rods and 6 million cones in the human eye. Rods and cones are key terms as well. So the description for rods is photoreceptors in the retina that function under low levels of illumination and do not give rise to colour sensations. And the description for cones is photoreceptors in the retina that function best in bright light and are differentially sensitive to red, green or blue wavelengths. The rods function in very dim light levels, being about 500 times more sensitive to light than the cones. However, rods are unable to generate a percept of colour. The retinas of some nocturnal animals, such as owls, contain only rods, giving them exceptional night vision, but no colour vision. The cones function best in bright light levels, and when there is more than one type of them, are able to generate a percept of colour. Some animals that are active solely during the day, such as pigeons, have only cones, so they can see in colour but have very poor night vision. Animals, like humans, that are active both night and day have a mix of rods and cones. In humans, rods are found throughout the retina except in the fovea, which is a small area in the centre of the retina that contains no rods, but many densely packed cones, and that is also a key term. Cones decrease in concentration the farther away they are from the centre of the retina, and the periphery of the retina contains mainly rods. Rods and cones send their messages to the brain via two additional layers of cells. The rods and cones have synaptic connections with bipolar cells, which in turn synapse with a layer of about 1 million ganglion cells, whose axons are collected into a bundle to form the optic nerve. The optic nerve is also a key term, and the description is the bundle of ganglion cell axons that carries information from the visual receptors to the visual area of the thalamus. Thus, input from more than 126 million rods and cones is eventually funneled into about 1.3 million output fibres that project from the retina toward higher visual centres. Figure 5.7b shows how the rods and cones are connected to the bipolar and ganglion cells. One interesting aspect of these connections is the fact that the rods and cones not only form the rear layer of the retina, but their light-sensitive ends actually point away from the direction of the entering light, so that they receive only a fraction of the light energy that enters the eye. The manner in which the rods and cones are connected to the bipolar cells accounts for both the greater importance of rods in dim light and our greater ability to see fine detail in bright illumination when the cones are most active. Typically, many rods are connected to the same bipolar cell. They can therefore combine or funnel their individual electrical messages to the bipolar cell, where the additive effect of the many signals may be enough to fire it. This is why we can more easily detect a faint stimulus, such as a dim star, if we look slightly to one side so that its image falls not on the fovea but on the peripheral portion of the retina, where the rods are packed most densely. Like the rods, the cones that lie in the periphery of the retina share bipolar cells. In the fovea, however, the densely packed cones each have their own private line to a single bipolar cell. As a result, our visual acuity, which is also a key term, or ability to see fine detail, is greatest when the visual image projects directly onto the fovea. Such focusing results in the firing of a large number of cones and their private line bipolar cells. Some birds of prey, such as eagles and hawks, have 2 foveas in each eye, contributing to a visual acuity that allows them to see small prey on the ground as they soar hundreds of metres above the earth. The optic nerve formed by the axons of the ganglion cells exits through the back of the eye not far from the fovea, producing a blind spot where there are no photoreceptors. You can demonstrate the existence of your blind spot by following the directions in figure 5.8, and that is on page 154. Ordinarily, we are unaware of the blind spot because our perceptual system fills in the missing part of the visual field. Figure 5.8 sort of looks like a long checkerboard with a dot on one side in one of the checkers and an X on the other, and it gets you to hold the screen or whatever you're looking at about 30 centimetres away and close your left eye, and then move closer to it, and then it says that the X will disappear and then it will come back into focus, and when it disappears, that's where your blind spot is. Well, when I held it away and closed my left eye, I can't even see the X, so that's where my blind spot is, but as I brought it closer, it came back into my vision. So cool. Visual transduction from light waves to nerve impulses. Rods and cones translate light waves into nerve impulses through the action of protein molecules called photopigments. That's a key term. Described as protein molecules within the rods and cones whose chemical reactions when absorbing light results in the generation of nerve impulses. The absorption of light by the photopigments produces a chemical reaction that changes the rate of neurotransmitter release at the receptor's synapse with the bipolar cells. The greater the change in transmitter release, the stronger the signal passed on to the bipolar cell, and in turn to the ganglion cells whose axons form the optic nerve. If a stimulus triggers nerve responses at each of their 3 levels, rod or cone, bipolar cell and ganglion cell, the message is instantaneously on its way to the visual relay station in the thalamus and then on to the visual cortex of the brain. However, rods use up all of the photopigment in normal daylight conditions, which means they do not operate in such conditions, being active only in low light conditions. Brightness, vision and dark adaptation. As noted earlier, rods are far more sensitive than cones under conditions of low illumination. Nonetheless, the sensitivity of both the rods and the cones to light intensity depends in part on the wavelength of the light. Research has shown that rods have a much greater sensitivity than cones throughout the colour spectrum, except at the red end, where rods are relatively insensitive. Cones are most sensitive to low illumination in the greenish-yellow range of the spectrum. These findings have prompted many cities around the world to change the colour of their fire engines from the traditional red, to which rods are insensitive, to a greenish-yellow in order to increase the vehicle's visibility to both rods and cones in dim lighting. Although the rods are by nature sensitive to low illumination, they are not always ready to fulfill their function. Perhaps you have had the embarrassing experience of entering a cinema on a sunny day, groping around in the darkness and finally sitting down in someone's lap. Although one can meet interesting people this way, most of us prefer to stand in the rear of the cinema until our eyes adapt to the dimly lit interior. Dark adaptation, also a key term, is the progressive improvement in brightness sensitivity that occurs over time under conditions of low illumination. After absorbing light, a photoreceptor is depleted of its pigment molecules for a short period of time. If the eye has been exposed to conditions of high illumination, such as bright sunlight, a substantial amount of photopigment will be depleted. During dark adaptation, the photopigment molecules are regenerated and the receptor's sensitivity increases greatly. Vision researchers have plotted the course of dark adaptation as people move from conditions of bright light into darkness. By focusing light flashes of varying wavelengths and brightness on the fovea, which contains only cones, or on the periphery of the retina, where rods reside, they discovered the 2-part curve shown in figure 5.9. And that figure is on page 155. The first part of the curve is due to dark adaptation of the cones. As you can see, the cones gradually become sensitive to fainter light as time passes, but after about 5 to 10 minutes in the dark, their sensitivity has reached its maximum. The rods whose photopigments regenerate more slowly do not reach their maximum sensitivity for about half an hour. It is estimated that after a complete adaptation, rods are able to detect light intensities 1/10,000th as great as those that could be detected before dark adaptation began. Colour vision. We live in a world rich in colour. The majesty of a glowing sunset, the rich blues and greens of a tropical bay, the brilliant colours of autumn foliage are all visual delights. Human vision is finely attuned to colour. Our difference thresholds for light wavelengths are so small that we are able to distinguish an estimated 7.5 million hue variations. Historically, two different theories of colour vision have tried to explain how this occurs. The first one is the trichromatic theory. Isaac Newton observed a fundamental aspect of our colour vision, namely that there is a distinction between the physical properties of the light and the resulting colour that we perceive. Specifically, that adding different colours together can generate other colours. He was particularly interested in white light, given that it was thought that it was pure and the different colours were produced by adding impurities to it. Newton showed that this isn't the case. He shone white light through a prism, breaking it down to its spectral colours, like a rainbow being formed by light hitting water particles in the atmosphere, and then recombined them to produce white light. Further, he found that the perception of white could be produced by adding only a few of the spectral colours together. This leads to the concept of metamers. Physically different stimuli can appear perceptually identical. Our understanding of how the visual system processes colour was developed using these metameric observations from colour matching studies. By 1800, it had been established that any colour can be produced by some combination of the wavelengths that correspond to the spectral colours red, green and blue. This fact was the basis for an important trichromatic, three-colour, theory of colour vision advanced by Thomas Young, an English physicist, and Hermann von Helmholtz, a German physiologist. According to the Young-Helmholtz trichromatic theory, there are three types of colour receptors in the retina. While individual cones are most sensitive to a particular wavelength, they are stimulated by most wavelengths to varying degrees. That is, cones are sensitive to a broad range of wavelengths, i.e. they have broadband tuning. The individual cones are most sensitive to wavelengths that correspond to either blue, green or red. Presumably, each of these receptor classes sends messages to the brain based on the extent to which they are activated by the light's energy wavelength. The visual system then combines the signals to recreate the original hue. If all three cones are equally activated, a pure white colour is perceived. Hear how my voice gets grainy the longer I talk for? Sorry about that. Although the Young-Helmholtz theory was consistent with the laws of colour mixture, there are several factors that did not fit the theory. Take our perception of yellow, for example. According to the theory, yellow is produced by the activity of red and green receptors, yet certain people with red-green colour blindness, who are unable to perceive either colour, are somehow able to experience yellow. A second phenomenon that posed problems for the trichromatic theory was the colour after image, in which an image in a different colour appears after a colour stimulus has been viewed steadily and then withdrawn. To experience an after image, follow the instructions for figure 5.11, which is on page 166. So the figure shows an image of the Australian flag with a green background, yellow and white stripes and black stars, and then you focus on a black spot and it will be different colours. And then it says, it is not immediately clear how the trichromatic theory can account for what you'll see. Opponent process theory. A second influential colour theory formulated by Herring in 1870 also assumed that there are three types of cones. Herring's opponent process theory proposed that each of the three cone types respond to different wavelengths. One type responds to blue or yellow, another to red or green, and a third to black or white. For example, a red-green opponent cell will give you a positive response to a red image and a negative response to a green image. You have experienced one of the phenomena that support the existence of opponent process if you did the exercise in figure 5.11. The after image that you saw in the blank space contains the colours specified by opponent process theory. The green portion of the flag appeared as red, the black as white, and the yellow as blue. According to opponent process theory, as you stared at the green, black, and yellow colours, the red, green, blue, yellow, and light dark sensitive cells that register those colours became fatigued. Then, when you cast your gaze onto the white surface, which reflects all wavelengths, each receptor responded with its opposing colour, that is, red, white, and blue respectively. Dual process in colour transduction. Which theory, the trichromatic theory or the opponent process theory, is correct? 2 centuries of research have yielded verifying evidence for each theory. Today's dual process theory combines the trichromatic and opponent process theories to account for the colour transduction process. The trichromatic theorists Young and Helmholtz were right about the cones. The cones do indeed contain one of 3 different protein photopigments that are most sensitive to wavelengths roughly corresponding to the colours red, green, and blue. Different ratios of activity in the blue, green, and red sensitive cones can produce a pattern of neural activity that corresponds to any hue in the spectrum. This process is similar to that which occurs on your TV screen, where colour pictures, including white hues, are produced by activating combinations of tiny blue, green, and red dots. Hering's opponent process theory was also partly correct, but opponent processes do not occur at the level of the cones, as he maintained. When researchers began to use microelectrodes to record from single cells in the visual system, they discovered that ganglion cells in the retina, as well as neurons in visual relay stations and the visual cortex, respond in an opponent process fashion by altering their rate of firing. For example, if a red light is shone on the retina, an opponent process ganglion cell may respond with a high rate of firing, but a green light will cause the same cell to fire at a very low rate. Other neurons respond in a similar opponent fashion to blue and yellow stimuli. The red-green opponent processes are triggered directly by input from the red or green sensitive cones in the retina. The blue-yellow opponent process is a bit more complex. Activity of blue sensitive cones directly stimulates the blue process further along in the visual system. And yellow? The yellow opponent process is triggered by a yellow sensitive cone, as Hering proposed, but rather by simultaneous input from the red and green sensitive cones. So according to the figure shown on page 157, it's saying that there are 3 kinds of cones, which are so that's trichromatic, and these cones have opponent process mechanisms. So blue wavelengths are short, green wavelengths are medium, and red wavelengths are long. So colour vision involves both trichromatic and opponent processes that occur at different places in the visual system. Consistent with trichromatic theory, 3 types of cones are maximally sensitive to blue, green, and red wavelengths, respectively. However, opponent processes occur further along in the visual system, as opponent cells in the retina, visual relay stations, and the visual cortex respond differently to blue versus yellow, red versus green, and black versus white stimuli. Oh, I swear it always rains when I decide to record the readings. Anyway, moving